everyone, and welcome to the micro-learning program from the Global Parkinson Genetics Program and Aligning Science Across Parkinson's. My name is Thiago Peixoto Leal, and I'm a postdoctoral fellow in Mata Lab at Cleveland Clinic, United States of America. Today, I will talk about local ancestry. We will divide this video in two topics. The first, genetic ancestry, and the second, local ancestry and phenotypes. According to the NIH website, genetic ancestry refers to information about the people that an individual is biologically descended from, including their genetic relationships. Genetic information can be combined with historical information to infer where an individual's distant ancestors lived. Using the genetic data from reference populations that works as a proxy to the ancestral population that may be one of the origins of the studied population, it is possible to infer what is the ancestry of an individual or population. Using population genetics methods, it is possible to infer the ancestry in different levels. The first is the populational ancestry, in which you measure the ancestry composition of your whole population, followed by individual ancestry, in which you measure the contribution of each parental population to a single individual. And using arrays with high density of variants or whole genome sequencing, we are able to infer the ancestry for each piece for each chromosome of an admixed individual. The local ancestry has been used in genotype-phenotype association studies in an effort to increase the power of the analysis performed in admixed populations. In the next slides, we will show some methods that are based or use local ancestry. The first example is admixture mapping. Unlike a genome-wide association study, in which you try to associate a specific variant with your phenotype, the admixture mapping tries to associate the local ancestry in the region with your phenotype. The admixture mapping can be performed in case control or case-only format. The case control design compares the local ancestry proportion between cases and controls in a region, so differences of the proportions of a specific ancestry in cases and controls may indicate that the variance associated with an ancestry in this region is increasing or decreasing the risk to develop the disease. The case-only design is based on the comparison of observed and expected ancestry to try to find regions enriched in a specific ancestry to cases. In 2023, Gouveia and collaborators showed that the European population, usually considered a homogeneous population, have some unappreciated subcontinental admixture. In this study, the authors concluded that genetic epidemiology studies may have spurious results for not properly accounting the ancestry. In this forest plot, you can see two different GWAS results, one for height and one for LDL, in which the authors tried four different models. The model 1, in which the model was phenotype explained by age plus sex. The model 2, that is the model 1, including the genetic relationship matrix. Model 3 and model 4, there are the model 2, including two ways to generate principal component analysis results. And model 5, in which they got the model 4 and included local ancestry. After including the local ancestry, the p-value changed drastically, showing that the results were biased by subcontinental admixture. Another approach is using local ancestry in order to increase the power in GWAS, as well as increase the power to detect ancestry-specific risk regions. There are several models that do this, and in this paper, you have a comparison of different models that use local ancestry to increase the power. Tractor is a method published in 2021 by Elizabeth Atkinson. This methodology uses the local ancestry, and it is a good method to find ancestry-specific risk loci. This is because of the way that the methodology works. Look at this example. The sample 1 has 4 SNPs in the VCF input, and the respective ancestry for each variant, the MSP input. In the first variant, he has a pink square, representing European ancestry, with a black dot, representing the risk allele. This merging between ancestry and risk alleles is done for all variants. After this information is used in a regression with 2 times n minus 1 variables, which n is the number of ancestries. The first variable, x1, is the dosage for European ancestry, or the number of pink squares with a black dot. The second variable, the x2, is the dosage for African ancestry, or number of blue squares with a black dot. The third variable is the number of European haplotypes, or the number of pink boxes the individual has. We do not include the number of African haplotypes for statistical reasons. Because the way that the tractor splits the dosage per ancestry, tractor has more power to detect ancestry-specific risk loci, but it is underpowered to risk variants in common to all ancestry or variants whose allele frequency is almost the same in all ancestries. So, some key messages. Ancestry connects genetics and society in fundamental ways, and there are studies that show that ancestry has an important role in several diseases. New methods to perform ancestry have been developed, and it is a pretty dynamic field. But the most important key topic is creating a very diverse cohort, as GP2 is important, but it also brings methodological problems. Local ancestry can be one of the resources that can help you. Thank you for listening, and I hope you enjoyed this video.